Okay, so chapter 35 deals with the digestive system, which, as we know, is made up of the teeth, tongue, the salivary glands, the lips, the cheeks, the pharynx, the esophagus, the stomach, the small intestine, the large intestine, the rectum, the anus, the liver, the gallbladder, and the pancreas. Okay, and the job of the digestive system is to mechanically and chemically break down the food you consume so that you can load it into the blood, in the case of the water soluble components of our diet, or into the lymph in the case of the lipid soluble components of our diet. Okay. And all of these have to work together in order for that to happen. Ingestion of food is a voluntary component. Propulsion of food and wastes from the mouth to the anus is carried out by something called peristalsis. All right. Let's write that down. Oop. There you go. Okay. Peristalsis. Okay. And these are waves of muscle contraction that propel the food through the tract until it gets all the way to the rectum, where waste matter is stored until elimination through the anus. Okay. Um, Smooth muscle does this. Okay. And there are autonomic components that trigger it, as well as hormonal. Okay. Secretion of mucus, water, and enzymes by the lining of the tract, very important as well. Okay. Mechanical breakdown of food particles. Again, that's going to be the property of chewing, peristalsis, and churning. Okay. This is voluntary. Put a V there. And these are involuntary. Okay. So again, both of these are autonomic functions. Autonomic nervous system. Okay. Chemical digestion of food particles is carried out by enzymes that are produced in the oral cavity, in the stomach, and in the small intestine. And in the colon, breakdown of our fiber is carried out by the microbes that live there. Okay, they have the enzymes to break down stuff like fiber, cellulose, hemicellulose, and so on. And we have absorption of ingested food, okay, into either lymph or blood. Elimination of waste products by defecation. And we have protection against infection because the whole thing, the whole tract, is a mucous membrane. Okay, you can see the mucosa here, all right, which has the simple columnar epithelium that runs from basically the stomach to the rectum, okay, in the digestive system. A lamina propria, which is areolar connective tissue with mucus glands and enzyme secreting glands in there, as well as brush border enzymes that are actually attached to the lining of the tract. A muscularis mucosa for local undulation of the mucosa to aid in contact with the lining of the intestine, and that aids absorption. Okay. Submucosa contain mucus glands, as well as an ample blood supply and a nerve supply. The lymph nodes, lymph nodules, are seen throughout, and that's to protect against ingested pathogens that survive things like bile and stomach acid so that they don't end up getting into the blood. Okay. The muscularis mucosa, I think we pointed out already. Okay. And then you've got the plexuses, right? There's a myenteric plexus and a submucosal plexus, which is part of something called the enteric nervous system, ENS. Okay. And the ENS is controlled by the ANS, even though it would operate on its own. All right. And also controlled by endocrine system. Okay, let's move this over. Okay, all right. The muscularis externa has an inner circular layer, inner, and an outer longitudinal layer, except in the stomach where there's an additional oblique layer that is deep to the circular layer. And the purpose, of course, is peristalsis, to push the food forward. And then on the very outside, you have either serosa or adventitia. In most areas, it's serosa, okay? Serosa, which produces a lubricating fluid that lets the tract move without inflammation and scarring, okay? And is a passageway for nerves and blood vessels and lymphatics going to and from the organ system, okay? The mouth is a reservoir for chewing and mixing food with saliva. Taste buds include for salty, sour, bitter, sweet, and umami. And then there's even a fatty taste that we're working on naming. Okay. All right. Remember that we're attracted to high calorie targets because of evolution. And that's one of the reasons that we like sweet and savory things, because they are high calorie. Okay, that's evolution, right? If we were attracted to high calorie food early in evolution, we lived long enough to breed. If we weren't, we didn't. Okay. Sour and bitter stuff was often toxic. Okay, which is one of the reasons that we're not attracted to it. Okay, those early ancestors that were, they died and they didn't live long enough to breed. Okay, so we're fighting millions of years of evolution with regard to the flavors we enjoy. Okay, salivary glands, there's three sets, right? A submandibular, which is under the mandible, sublingual, under the tongue, and a parotid, which is in front of the external ear. Saliva is what's produced. It's water with mucus, sodium bicarbonate, chloride, potassium, and salivary alpha amylase for carb digestion, and is controlled by sympathetic and parasympathetic fibers. Okay, sympathetic stimulation reduces saliva production, and parasympathetic increases it. Okay, swallowing is essentially a combination of voluntary and involuntary action. Okay, basically the idea here is that we propel food from the oral cavity into the pharynx, and then from the pharynx into the esophagus. Okay, and once that happens, peristalsis takes over to push the food ball, which is called a bolus, through the cardiac sphincter, which is the doorway into the stomach, where we liquefy the bolus with the gastric secretions and turn it into a liquid called chyme. The upper esophageal sphincter keeps air from entering the esophagus during respiration, while the lower one prevents regurgitation from the stomach and caustic injury to the esophagus. Okay, so let's look at swallowing.
Swallowing, or deglutition, is the process by which food passes from the mouth to the stomach. This movement is controlled by the nervous system and involves both voluntary and involuntary muscle contractions. Before food is swallowed, it is mechanically broken down in the mouth by chewing or mastication. The salivary glands secrete the enzymes lingual lipase and salivary amylase. Salivary amylase begins the chemical breakdown of starch, carbohydrates, in the mouth. The water content of saliva helps bind particles together to form a bolus that can be swallowed. Swallowing occurs in 3 stages. The first is a voluntary phase called the buccal or oral transit phase. Here the tongue moves upwards and backwards against the hard and soft palates to push the bolus into the oropharynx. Next is the pharyngeal stage, in which involuntary movements push the bolus through the pharynx and into the esophagus. Movement of the bolus stimulates receptors in the oropharynx, which send impulses to the deglutition center in the medulla oblongata and the lower pons of the brainstem. The brain signals the soft palate and uvula to close off the nasopharynx, and the epiglottis to seal off the larynx. This prevents the bolus from entering the respiratory tract. The upper esophageal sphincter relaxes to allow the bolus to move into the esophagus and contracts to reduce backflow into the pharynx. In the final esophageal stage, the bolus is pushed onward by peristalsis, a progression of involuntary wave-like contractions of the circular and longitudinal smooth muscles of the esophagus. As the bolus approaches the end of the esophagus, the lower esophageal sphincter relaxes and the bolus moves into the stomach. So, in summary, food moves from the mouth through the oropharynx and esophagus and then into the stomach.
Now, your stomach is an unsung hero of the digestive system because it allows you to do discontinuous feeding, okay, which means that you can eat a meal and go on with your life, all right? Discontinuous feeding. Okay. If we couldn't do this, we would live the life of an earthworm, which is what? Eating dirt, pooping, reproducing, and dying, okay? Which is pretty much every day for an earthworm, all right? So the stomach allows us to do this. It's a hollow organ that produces gastric juice and an alkaline mucus. The gastric juice contains hydrochloric acid and an enzyme called pepsin. Pepsin begins protein digestion. Okay, this is the first place where protein digestion happens. Starch and lipid digestion actually began in the oral cavity with lingual lipase and salivary amylase, but those get destroyed upon entering the stomach because of the low pH. Okay, the boundaries of the cardiac orifice, the pyloric sphincter, and the pylorus itself. The cardiac orifice is the doorway into the stomach, and the pylorus and the pyloric sphincter are the doorway out. Okay, so they attach to the duodenum while the cardiac orifice attaches to the esophagus. The areas of the fundus, body, and antrum, and you can see them here. Okay, we told you that the stomach has an extra layer of smooth muscle, an inner oblique layer, to aid in churning. All right, the idea there is to liquefy the bolus so that it can be pumped into the duodenum in tiny squirts at the same time that the gallbladder contracts and brings in to the duodenum both bile and pancreatic juice at an area called the hepatopancreatic ampulla, which is surrounded by a little smooth muscle doorway called the sphincter of Oddi, O-D-D-I. Okay. Blood is supplied from the celiac artery to the stomach, which also has rugae, as you can see indicated down there, along its inner lining. These are just folds that let the stomach expand as food enters it. Okay. And of course, there's a serosa on the outside of the stomach, just like there is along most of the rest of the tract, and it lubricates and suspends the stomach in place so it doesn't twist, buckle, or kink, as mesentery does for the rest of the digestive system. Okay. There is the fundus up top, right? The body of the stomach, the pylorus, okay, and the pyloric antrum, three main parts of the stomach. And there you can also see the lesser curvature, which is attached by the lesser omentum to the underside of the liver, and the greater curvature, which is attached to the greater omentum, which is also attached to the transverse colon. Okay, the omenti are connective tissue layers, which again provide avenues for lymphatics and blood vessels and nerves to go back and forth from the organ system. And in the case of the greater omentum, fat storage. Okay. Swallowing causes relaxation facilitated by gastrin and CCK, which is the short name for cholecystokinin, CCK. Okay. Motilin increases peristalsis, secretin decreases peristalsis. Okay. And gastric mixing and emptying are provided by the action of those three smooth muscle layers, resulting in movement towards the antrum and toward the pylorus, and retropulsion if the pyloric sphincter isn't open. Okay, the rate is dependent on the volume, the osmotic pressure, and the chemical composition of what's in the stomach. Gastric secretion has several phases: the cephalic, gastric, and intestinal phase. The cephalic phase is stimulated by the thought, smell, and taste of food. This is why your stomach growls when you smell donuts. Okay. The gastric phase is stimulated by stretching of the stomach, and the intestinal phase is stimulated by histamine and digested protein entering the duodenum. Okay. Stimulation of gastric secretion happens as a result of the act of eating, but also the secretion of gastrin from the enteroendocrine cells in the stomach. Okay. Specifically G cells. All right. There are paracrine pathways as well. All right. Acetylcholine is a stimulus, and chemicals like ethanol, coffee, and protein are a stimulus as well. The stomach secretes large volumes of gastric juice, which includes mucus, acid, enzymes, hormones, intrinsic factor, and gastroferrin. Okay, intrinsic factor is to allow B12 access to the blood. Okay, and that's important so that you can make blood. All right, gastroferrin binds iron. Okay, enzymes chemically break down our food. Okay, hormones coordinate activity in the tract. Acid sterilizes food. And chemically breaks it down. Okay, and mucus protects the gastric lining from acid so that you don't carve a hole in it as easily. Okay, gastric secretion. Acid is secreted by parietal cells. This is hydrochloric acid. It's a sterilant. It aids in protein breakdown. It activates pepsinogen, which is the inactive form of pepsin, which is a protease. Okay, pepsin is secreted by Chief cells. It's a proteolytic enzyme and begins protein breakdown. And mucus, of course, stimulated by prostaglandins, protects the stomach from its own secretions. It's an alkaline mucus, and it also helps to lubricate material in the stomach. All right, and you can see here the different cell types: the enteroendocrines that make the gastrin, and the enterogastrones, the parietal cells. All right, and the chief cells, and the mucous neck cells. Okay, now you can see again the same basic layers, right? Mucosa, submucosa, muscularis mucosa, right? And then muscularis externa, and then serosa on the outside. Okay. After we liquefy the food in the stomach, it becomes chyme, and the chyme has to be squirted into the duodenum. All right. The small intestine has three segments: the duodenum, which is the first few inches; the jejunum, which is about 8 feet; and the ileum, which is about 11 feet. The ileum joins the cecum at the ileocecal valve. Okay. The peritoneum is the membrane that surrounds the organs of the abdomen and pelvic cavity. All right. There is a visceral peritoneum that clings to the organs, and a parietal peritoneum that clings to the chamber that the organ is in. Okay. And they all are in the peritoneal cavity. Okay. The peritoneum, remember, has several roles. It latches the organ system in place so it doesn't twist, buckle, or kink. All right. It's a pathway for nerves and blood vessels and lymphatics to and from the organ system. All right. And it produces a lubricating fluid so that when the organ system moves, it doesn't irritate, inflame, and scar itself. The duodenum is supplied by the gastroduodenal artery. The jejunum and ileum supplied by superior mesenteric. It's innervated by the enteric plexus. There's two sets: the myenteric or Auerbach plexus that talks to the muscularis externa, and the submucosal plexus, which talks to the muscularis mucosa, okay, as well as gland targets. The two layers of smooth muscle in the small intestine are the inner circular and outer longitudinal layers, okay. Absorption in the small intestine is paramount, and that's one of the reasons that many modifications are seen in the small intestine that permit increased surface area. Okay, one is the folding of the pipe on itself. The other are ridges and grooves within the pipe called plicae circularis. The other are the villi that protrude from the plicae circularis, and on their surface, simple columnar epithelia with microvilli on their apical surface. Okay, you could see that there if you look at the brush border of the lining of the small intestine. And then, of course, embedded within the villi are the lacteals that absorb dietary fat, and the enteric capillary networks that absorb the water soluble component of our diet once it's tiny enough to pass through, right? So proteins have to be broken down to amino acids, and complex carbohydrates into simple sugars, and of course fats into fatty acids and glycerol, and of course along with that is the absorption of water soluble vitamins and minerals into the blood supply, and absorption of fat soluble vitamins A, D, E, and K into the lacteals. Okay, now where does all that blood go? We're going to find out. It goes into the back of the liver through the hepatic portal vein and mixes with blood from the hepatic artery. Okay, we initiate protein breakdown in the stomach, and sterilization is facilitated by the action of hydrochloric acid. Okay, um, we can carry B12 across the lining of the stomach as well as certain other drugs. Okay, such as alcohol and aspirin. All right, pepsin promotes protein breakdown. This continues in the proximal small intestine. Pancreatic enzymes, intestinal enzymes, and bile salts break down carbohydrates, proteins, and fats, and the nutrients are absorbed by either active or passive transport. Okay. Intestinal motility is the product of smooth muscle action, which is initiated by local and long range reflexes facilitated by the autonomic nervous system via the enteric nervous system as well as the endocrine system. Okay. Haustral segmentation, peristalsis, the iliogastric and iliointestinal and gastroileal reflex are all examples of these. Okay. The ileocecal valve is simply the doorway between the small intestine and the large intestine. The cecum is the first part of the large intestine, is a pouch that gets the chyme from the ileum, and off the bottom end of the cecum you can see the appendix, which is a site for bacteria that recolonize the colon to remain in the event that they're needed to repopulate the colon if your normal gut bacteria get wiped out. It occasionally can get plugged, and gas produced by the bacteria that live in there can cause the blind-ended sac there to swell, and it can potentially rupture and break contained between the digestive system and the abdominal pelvic cavity, leading to peritonitis, septicemia, and death. Okay, so it's nothing to laugh about. The diagnostic is extreme pain at McBurney's point, which is halfway between the crest of the right ilium and your navel. Okay. The colon has several parts after the cecum: the ascending colon, the transverse, the descending and sigmoid colon, ending at the rectum and the anus. You can see the pouch-like haustra produced by contractions of the teniae coli, which are the longitudinal smooth muscle layers in the colon. Okay. And then there are the blood supply, right? The descending and sigmoid colon, the rectum, and the anus are supplied exclusively by the inferior mesenteric, and then everything upstream of that, from the transverse on back through the small intestine up to the stomach, is superior mesenteric artery. Okay, you can see that the hepatic portal vein drains not only the large but also the small intestine and the spleen and the stomach. Okay, and all of that, and the pancreas, and all of that goes into the back of the liver, mixing with blood from the hepatic artery. Okay, go move. There you go. Yeah. The ileocecal valve is the doorway between the large and small intestine. The end of the trip, there are two sphincters. There's an internal and an external anal sphincter. The internal anal sphincter is smooth muscle. Okay, smooth muscle. Write that down. and the external skeletal muscle, which you control consciously. Both have to be relaxed in order for defecation to happen. We talked about the Taenia coli and the Haustra. Okay, so we know what those are about already. Motor and secretory activity is regulated by the enteric nervous system, the ENS. Okay, the ENS, which is controlled by the ANS and also controlled by the endocrine system. Okay. Vagal stimulation increases rhythmic contraction of the proximal colon. The blood supply is derived primarily from branches of superior and inferior mesenteric arteries. And you can see here how the movement operates, right? It massages the fecal mass, it recovers water and electrolytes. Peristaltic movements promote the emptying of the colon. The gastrocolic reflex causes the fecal mass to pass rapidly into the sigmoid colon in the rectum, and the defecation reflex is stimulated by the movement of feces into the sigmoid colon and rectum. Um, your immune system works with your digestive system by recognizing our good bacteria, our good flora, and responding to potential pathogens. Okay. Mucosal secretions in the GI contain IgA antibody, lysozyme, and lactoferrin. Okay. Pannus cells produce defensins and antibiotic peptides. Peyer's patches, which are just beneath the epithelial lining, right, which are made up of lymphocytes and macrophages, produce immunoglobulin as a component of gut-associated lymph tissue in the small intestine. So remember that when we're talking about a mucous membrane, the inner lining that is around the lumen is going to be simple columnar epithelium running from the stomach all the way to the rectum. Okay. And then beneath that is loose connective tissue with lots and lots of white blood cells in there. And particularly when we get to the ileum, all right, you get Peyer's patches, which are significant amounts of lymphocytes and macrophages that are there primarily to protect against bacteria refluxing from the large to the small intestine and potentially breaching contain of the epithelial layer and getting into the connective tissue. Okay, so our gut bacteria are good in the colon, but bad in other places. And that's why we have to keep guard and not let that stuff get into the blood supply. Multiple factors are going to affect normal composition of bacterial flora, including genetics, diet, environmental components, drugs you take. Alerting the immune system to protect against infection is critical. It's one of the purposes of the appendix. Okay. Your intestinal tract is sterile at birth, but colonized in a few hours. The number and diversity of bacteria decrease as you age. Bacteria in the stomach are sparse because the acid is sterilant. Okay. Low concentrations of aerobes in the duodenum and jejunum, primarily because of the action of bile, which is a detergent, okay, as well as the fact that stuff is constantly moving and the presence of the antibodies cuts down on growth as well. Anaerobes distal to the ileocecal valve are found, but are not proximal to the ileum, okay. Splanchnic blood flow provides blood to esophagus, stomach, small and large intestine, liver, gallbladder, pancreas, and spleen, and is an important reservoir of blood volume. And you can see here what I like to call mission critical for the digestive system. Okay, you've got the liver in the upper left there, covering up portion of the stomach. Okay, you can see behind the liver is the gallbladder, which receives bile via the common hepatic duct via and into the cystic duct, and then the gallbladder concentrates that bile, so it's a better emulsifying agent. Okay, you see the pancreas behind the stomach, the spleen lateral to the pancreas. Okay, and you can see where the common bile duct and the pancreatic duct fuse right there at the duodenal papilla, sometimes called the hepatopancreatic ampulla, governed by the sphincter of Oddi. Let's spell that. Okay. What happens when we want to release pancreatic juice and bile at the same time that chyme comes from the stomach into the into the duodenum, right, is that two things happen, right? CCK is a hormone that is going to cause the gallbladder to squeeze at the same time that it causes the pancreas to make pancreatic juice. And then there's another one called secretin, okay? Secretin, which causes the pancreas to produce bicarbonate. Okay. The other thing that CCK does is it causes the sphincter of Oddi to squeeze. Okay. So let's draw that in. Okay. There it is. Causes the sphincter of Oddi out here. Okay. To squeeze. Sorry, to open. Write that. Okay. We'll draw this here in blue now. Okay. Opens the sphincter of Oddi at the same time that we squeeze the gallbladder, right? And then all of that bile, pancreatic juice gets in this duodenum at the same time as the the payload of chyme from the stomach in little squirts. Okay. So that's why I call this mission critical for the GI. The liver has several lobes, a left, a right, a caudate, and a caudate and a quadrate. Sorry, they're shown down below. The falciform ligament is essentially going to separate here the right from the left lobe on the front. Okay. Falciform ligament there. Okay. The round ligament. Okay. Which is right along here is a remnant of the ductus venosus. Okay. And then the coronary ligament is going to be right near, it should be right near the heart. Looking for it here. Da, 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 I'm not seeing the coronary ligament. Okay. But I'm going to tell you that it's up here somewhere. Okay. Okay. The liver is unique because it has a couple of interesting properties. It can regenerate, and it has immune privilege. Okay. Regenerate means you can lose up to 80% of your liver, it'll grow back, which is pretty neat. Immune privilege means that if you're type O, you can donate a lobe of your liver to anybody that needs it. Okay. You only have to match blood type and not tissue type. Okay. You can see the blood supply, right? Hepatic artery supplies blood to the liver along with the hepatic portal vein. All right. That blood percolates through the liver. Let's find those here. There's the portal vein. Okay. That blood percolates through the liver, and then the hepatic vein dumps into the inferior vena cava, the treated blood, and this heads, of course, to the heart. Okay. They're not showing the hepatic artery here, but the hepatic artery enters the back of the liver at the same spot that the portal vein does. All right. So basically, nutrient rich blood is going to flow from the the large and small intestine, the stomach, okay, pancreas and spleen, right into the back of the liver, mixed with hepatic arterial blood, and that percolates through the liver and it comes out the hepatic vein into the inferior vena cava right there. Okay. Now, you might wonder, okay, so what all happens to that blood? What's going on with that blood going through the liver? Well, liver does a bunch of stuff, right? Makes plasma proteins. Okay. Decontaminates blood. detoxifies blood. Recycle RBCs. Okay. Make bile. Okay. Adjust the nutrient content of blood. Okay. That's why if you lose your liver, you lose your life. That's why the first four words of the word liver spell live. That's not an accident. That was done on purpose. Okay. Fortunately, it grows back, which is nice. Okay. The liver lobules are made up of groups of hepatocytes, which are hexagonally shaped, as are the lobules. The sinusoids connect the triads of the liver lobules to the central vein. Biocanaliculi collect bile from the hepatocytes and eventually dump it into the common hepatic duct, which empties into the cystic duct, which empties into the gallbladder. Common bile duct is going to connect to the cystic duct and empty into the pancreatic duct, and both of them are going to empty into the hepatopancreatic ampulla. Here they're calling it the ampulla of Vator. It's also called hepatopancreatic. Okay, and of course the doorway out is the major duodenal papilla, which is controlled by the sphincter of Oddi, which is basically the doorway into the duodenum. For pancreatic juice and bile. Okay. Kupffer cells are macrophages, fixed macrophages in the liver that remove pathogens in cancer cells. Stellates are contractile in liver injury, contain retinoids and remove foreign substances from blood. Natural killers produce interferon gamma and are important in tumor defense. And the disc space drains interstitial fluid into the hepatic lymph system. Okay. Bile is basically a detergent. It has a green appearance to it because of the presence of bile salts, which contain bilirubin, which is green. Okay. There's also cholesterol in there, as well as electrolytes and water. It's formed by the hepatocytes, secreted into the canaliculi, and enters the enterohepatic circulation, in which it leaves through the bile duct into the duodenum, and then it re-enters via the ileum back into the drainage from the small intestine into the hepatic portal vein. Okay, so a portion of it cycles and cycles and cycles again. Okay, so there is your enterohepatic circulation. Okay, the components of bile, there's an acid dependent and independent component. The bile salts include cholic salts and canic salts. Secondary bile salts, deoxycholic and lithocholic. Primary and secondary bile acids together form the bile acid pool. Bilirubin is a breakdown product of red blood cells that gives the bile a green, black color and produces the yellow tinge of jaundice. Bilirubin can either be conjugated or unconjugated. Conjugated bilirubin can be excreted. Unconjugated bilirubin, not so much. Okay. Urobilinogen is one of the breakdown products of bilirubin that ends up in the urine. That's why your urine is yellow. Okay. What else does the liver do, right? Well, we talked about
Mr. Richard, can I ask you a quick
yes, yes,
question?
go ahead. Yeah, go ahead.
What differentiates between the unconjugated and conjugated bilirubins?
Okay, so what goes on in the liver is that there's an enzyme called cytochrome P450.
Right.
Okay. And the job of cytochrome P450 is to attach water soluble groups to lipid soluble compounds so that they can eventually, when they get to the kidneys, be excreted into the urine. Okay. Otherwise, they would remain lipid soluble. So when we're talking about conjugated versus unconjugated bilirubin, that's what we're talking about is these large water soluble groups that are attached to the bilirubin in order to help get it excreted. Okay. Because eventually your bilirubin leaves here and here. Here is urochrome. And here is stercobilin, which is brown. Okay. So it doesn't allow it to accumulate. Okay. This is one of the reasons why lipid soluble stuff puts a terrible load on your liver, because you make it work so hard with cytochrome P450 that sometimes you damage the liver cells. Okay. Another thing that the liver does, of course, is it detoxifies alcohol and recreational drugs using the same system for recreational drugs, and using a pathway to oxidize alcohol that produces acetaldehyde as an intermediate, which of course is very, very chemically similar to formaldehyde, which damages living tissue. So this is one of the reasons why alcoholics lose liver function, is because they build up large amounts of this intermediate, and it basically destroys their liver cells. Okay. Okay. Your liver stores blood.
Thanks.
Yes, you're welcome. Stores blood, makes clotting factors, makes complement, makes albumin, all of that. Okay. Very important for normal function of blood. Metabolism of nutrients, fats, proteins, carbohydrates. The liver helps with all of this because it can do transamination and deamination. Okay. In transamination, we steal an amino group from one molecule and slap it onto another. In deamination, we simply remove it and it becomes either urea or ammonia. Okay. It does metabolic detoxification and stores minerals and vitamins. And we're talking here minerals and vitamins, um, vitamins, the fat soluble types. Okay. And then minerals um, in the form usually of being complex with proteins. The gallbladder just hangs on the back of the liver and stores the bile the liver makes so it becomes more concentrated. Um, it holds about 90 mil of bile, begins to contract half an hour after eating under the influence of vagus nerve and CCK, okay, which also, remember, makes the sphincter of Oddi open up. The exocrine pancreas is composed of acinar cells that secrete bicarbonate and digestive enzymes, okay. The pancreatic duct is going to fuse with the bile duct and empty into the hepatopancreatic ampulla. Here they call the ampulla vater. Either is acceptable. Arterial blood supplied by branches of celiac and superior mesenteric arteries. Venous blood leaves through tributaries to the portal vein and the splenic vein. Okay. The secretions are isotonic and have potassium, sodium, bicarbonate, chloride in them. Enzymes hydrolyze protein, carbs, and fats, including trypsin, chymotrypsin, carboxypeptidase, and elastase, and are secreted in inactive forms. Trypsin inhibitor prevents the activation of proteolytic enzymes while they're in the pancreas, so that we don't digest the pancreas prematurely. Okay. And enterokinase is made by the duodenal mucosa and activates inactive enzymes as well. Okay. All right. So let's pause here for 15 and we'll get into the next chapter. We'll keep these. Okay. And then we'll do this. We will, where's the more? There you go. I want to, here, come on, stop.